Okay, well, if you are looking for BMS 212, you're in the right place. I'm Professor Leonard. A few minutes ago, I was just reflecting on how many ways there are to screw up. Right, so when I came over here, I had my laptop and my iPad and this, and that. I had everything. And I just realized a few minutes ago that I do not have my lecture notes. So I would just ask for a little bit of grace on this first day. I think that I've got it okay, but if I leave anything out, we'll talk about that next time. I am recording to a Zoom meeting. So that's an option, perhaps, that if you want to jump on the Zoom meeting sometime instead of coming to class. Again, I'm recording all the lectures. You can come on in. There's lots of chairs up here yet, so just come on in. Or not. Okay, so that's an option. There's supposed to be 140 seats in here, and there should be only 120 of you. But yeah, we'll be pretty full on exam days. Okay, so don't be afraid to jump in. It's a little bit hard to ask questions in this kind of room. It's hard to hear each other. If you do have trouble hearing me, make sure you let me know that. I have another microphone system that I was planning on using, but I don't know how to disconnect the room one. So I'll figure it out if that's necessary. So let me know if you have trouble hearing or if there's any other distractions. That was a constant problem last semester, basketballs or music or whatever. We had to keep, reminding the rec center to keep those distractions to a minimum. Okay, so usually I start out lecture with a little spiel of trying to convince you why you should be interested in microbiology. If you've been conscious in the last year, then hopefully you are at least somewhat interested in microbiology because there's a little virus that's pretty much dictated everything we've done in the last year. It's the 1-year anniversary of the first case that we for sure described in the US. So hopefully after taking this class, you'll be a little bit better informed about decisions that you have to make, all sorts of public health decisions, vaccines, all sorts of things. And a lot of you may have already been presented with the choice to be vaccinated or not, or at least some of you that work in a healthcare setting. So I really wanted to talk about vaccines, at least just for a couple of minutes, right from the get-go. We'll talk about the science of vaccines later on in the course in April when we talk about immunology and why vaccines work, and we'll talk about the COVID vaccine and how the process that it had to go through to be vetted to be used. But I wanted to talk a little bit, just briefly here, about a family friend. Her name's Kate. And if you're from the Jenison-Hudsonville area, you may know Kate and her family because her family for many years has raised money to try to cure the incurable genetic disease that she and her sister have. It's a disease called AT, and it's a neurodegenerative disease. It affects a lot of aspects of her health, but it's also made her immunocompromised. So there's some aspects of her immune system that don't function as they should. Her younger sister died when she was 11 of influenza. So it's a pretty serious disease. Kate has beat the odds. She was only supposed to live 12 years, and she's 23 now. So she's doing great, but COVID, if she gets the COVID vaccine, her body is not going to respond like it should, and she will not be protected even if she gets vaccinated. So I think this is the biggest argument for all of us. I hope that when you have the choice whether to be vaccinated or not, I hope that you'll say yes. If enough of us get vaccinated, then people like Kate will be protected. So, and even not thinking about, people that are vulnerable in our population, but just thinking you yourself, there's a 0.001 chance that you'll have a reaction to the COVID vaccine, and you might be uncomfortable for a couple days. Okay, think about the chances if you get COVID the chances that you be very sick or maybe have some long-term issue to do with COVID. So just even looking at yourself, odds are you're going to be better off getting the vaccine. So I'd be happy to discuss that with anybody that wants to talk about that anytime. Okay, down to a little bit syllabus discussion. Like I said, my name's Susan Leonard. My office is in Henry, right down the hall from the labs. So if you know where the labs are, I'm right down the hall from that. The office hours, I am planning on being both virtual and open door at the same time. We'll see how that works. I might need to mute somebody on Zoom for a little bit or something like that. But I had my Zoom link open today from, I was open from 10:30 to 11:30. I had that wrong. So I'll do that from 10 to 11. I made a tab on Blackboard. I think it says syllabus, schedule, and Zoom link, or something like that. So it should be easy to find. It's going to be the same number every day for lecture and every day for the office hours. Okay, don't be afraid to ask if those office hours don't match up well with your schedule. I'm available on Fridays. I'm available lots of other times as well. Okay. Just a little bit about me. There's my happy family there, picture last summer. My husband, Dr. Leonard, teaches in chemistry. I know that some of you know him. I am technically not a doctor. So if you call me Dr. Leonard, I answer to that, but Professor Leonard is probably more appropriate given my education there. So we have 3 kids. They went to Jenison High School. The youngest there with his pandemic haircut, he goes to Michigan. He's an engineering student. Our middle child, she has autism and she goes to Young Adult Services in Hudsonville. And she also works at a company that makes yachts. So we kind of laugh about that, that she's helping build big boats a couple days a week, carpet and insulation and that kind of stuff. And then my oldest, she's in her first year of medical school. So I'm pretty familiar with that journey. If any of you want to talk to me about that. The school that she goes to in Washington, D.C. Does not require, they don't charge a fee to apply. So I would encourage you all to apply to this school. It's called USU, or Uniformed Services University of Health Sciences. It's affiliated with the military. She gets paid money to go to medical school. So what kind of deal is that? It's the best deal you have. She has to serve a few years when she's done, but she loves it. She's just in a good place right now. So I'd love to chat about that with any of you. All right, I see that about 70 of you have already signed up, are into the course on mastering. I did manage to copy this code wrong on the syllabus. So it's like I checked that so many times. This is the correct number. It's a 7 on the end there instead of a 4. Hopefully that didn't cause too many of you any consternation. But that's the code you'll need to get signed up. So that code will give you access to the online homework, which is about 10% of your grade. So you're not going to want to ignore that. It has access to the e-text. It has access to lots of other study helps and animations and practice quizzes. I think it's a pretty good resource. Okay, so let me know if you have any questions about ordering that. If you do want a real textbook, this is what it looks like. This is the 6th edition. The 5th and 4th edition are really, really close to this. In fact, some of the pictures that I show in class are from old editions. So those are good too. The page numbers are just slightly off, which really shouldn't be too much of a problem. Okay. You can also order that in loose leaf for like $45, and that's, they give you the option to do that. Okay, hopefully we will be in person for all our exams. Okay, I did have to give a couple of exams by respondus last semester, and that's what we would do if for some reason we had to close the university. So if we have to do that, I'll give you some practice if you haven't had to do that before. But Hopefully not. Okay. Let's talk about grading. Every other time that I have taught this class, and this is, I'm starting to lose count how many times I've taught this. I think it's like the 8th or 9th time I've taught this class. I've taught lab for about 15 years. So I've been here a little while. Every other time I've taught this class though, I have given 4 exams rather than 3. The reason I went down to 3 exams this semester, even though that makes them a little bit more pressure on each one, the problem was when I looked at my syllabus from last semester and made up my syllabus for this semester, I had 3 less class periods. So that's 3 hours and 45 minutes less this semester with you in class than I had last semester. So I get one of those days back by going to 3 exams rather than 4. So this is what I spent my break, trying to decide. I trimmed a few things back that I thought maybe were a little less essential, talked to some other people, but I know that I didn't trim enough. So we may get to a spot where I will say, okay, I'm gonna have to post this 15 or 20-minute lecture with a little bit more content. And I'll try not to do that like the night before the exam or anything like that. I'll try to give you lots of heads up, but there may be things that I think are important enough that I don't want to skip. So just kind of, that's where I'm at with that. I always like to make the final exam worth a little bit more. A year ago, when we had to shut down because of the pandemic, I thought it'd be nice to students and make the final exam worth less. And lots of people were up in arms about that because they wanted those extra points. If you start to do better as the course goes on, you want that final weighted a little bit heavier to reflect that maybe you had a bad first exam or something like that. Okay, I made the homework worth the same amount as what I would have done if there was 4 exams. So what that means is the homework is worth a little bit more than what I've had in the past, like 12% versus like 9%. Okay, and that's good because you all should get all those homework points with just a little bit of effort. Okay, so the way I have the homework set up, and refer to the syllabus for a little longer explanation of this, is that there's a certain number of possible homework points. It's actually a little less than 516 because I tried to take out the homework questions that were over things that I wasn't going to talk about anymore in class. Okay, so there's a certain number of homework questions over the whole semester. And if you get 80% of those correct, then I will give you the full homework points. The reason, that's not the way I've always done it. The first time I taught, it was, whatever homework point you got, then you got credit for that. So maybe you ended up with 56 or 55 homework points. But I found that students were spending hours and hours and hours doing those homework just to get every little point. And it really, to me, defeated the purpose. The homework is there to give you some practice. Okay, so, if there's a question that's kind of off the wall, you might just guess over it, or, you didn't, listen to that whole lecture or whatever. So you might guess over a few, but as long as you're getting about 80% over the whole semester or just above it, then I'll give you those full points. Okay, if where students run into problem is if they save the homework till right before it's due, and then they end up not having time to finish it. And then they maybe finish the semester with 70% of the homework points. So then you would get 70% of 60. All right, so you're giving up some points that are really just, they're just there for you to do. Okay, so again, I think the homework is good review. It helps you see things again, all right? So you watch the lecture, you do the homework, you look at it in the book a little bit. Like, you need to see things several different times and maybe in a couple different ways to really start to get it to click in your head. That's the way I am, at least. So it gives you a chance to see things several different ways. There are some homework questions that are like case studies or things like that that are meant to just try to get you a little bit more interested or to, see some applications. But I try to only ask you to do homework questions over things that we have covered in class and over things that you'll be tested over. So Occasionally there might be a question that has like 5 parts, and a couple of the parts are things we didn't talk about, but I couldn't really tweeze those out. So, okay. All right, so that's the homework. Of course, you're probably thinking, you might have heard horror stories about this class. Hopefully not too many, but hopefully you're wondering, how can I do well in this class? So, you know, the literature says, okay, you do the book reading before lecture, and then you kind of know what I'm talking about in lecture, and then that's the second time you've seen it. This is, so I understand that you have other classes and you may have a job and you have other things. So this is like really a best case scenario. And really those pages that I have posted on the syllabus, there might be some material kind of in between there that, you know, I'm not going to be covering in class, that I'm not going to be asking you about. So, you know, you may be reading some things that aren't absolutely necessary. I anticipate that most of you will use the textbook more as a reference. So maybe we're covering something in class like viral replication. It's a little bit more complicated, a little more confusing. I need to see that again. So you read about it in the book, and that again helps you kind of understand what's going on. So there will be certain parts, I'll tell you when we get to them, and I'll say, look, it's really going to help you if you read the book about this section. This is more complicated. You're going to want to read it. Other words, it's a good reference, especially, you know, looking up homework questions, things like that. Okay, I did post PowerPoints probably today. It might get us through the next lecture. So you'll have to see what works for you, whether you want to print those out, whether you can take notes right on it. That would be awesome, because that's the way I'm going to be doing it in class. You might be able to type notes. You have to see what works for you. But you will need to take additional notes outside of just what I post up there. Okay, try not to do this death by PowerPoint. I try to make sure that you get involved a little bit. Okay, there is a tab on Blackboard that says Learning Objectives. Okay, that's about 8 or 9 pages. I would suggest that you print those out right from the get-go. Some of these I've made up myself. A lot of them I've just taken straight from the textbook, but I've picked and chosen. It's not all the ones in the textbook are right here. So what I do when I make an exam, I look at the objective and then I make up a question that fits in that objective. So I know some of these are vague. Okay, some of them are pretty specific, but that I think should be helpful to you. I tried to put some terms that I thought might be new. I tried to boldface them on the learning objectives. Because one of the things that I like to stress is terminology. Any biology course, you really got to be fluent in the terminology to be able to, you know, read a paper, a research paper. If you have to stop and look up everything, you're just not going to know what it's talking about. So don't be afraid to stop and ask me if I'm using some term that you don't know what I'm talking about. Sometimes you're going to have to, you know, obviously review those, but that's something that we'll stress. Okay, and like I mentioned before, some students think, oh, I'm going to do the homework a day before the test, and that will help me review for the test. Just give yourself plenty of time. You know, after today, you should be able to do the homework from chapter 1. Should be a little easier because I've already, you know, talked about it. You won't have to look every little thing up. You know, don't wait till 2 weeks from now to do the homework from today. I think you're going to, I think that strategy is going to work better for you. Okay. Questions? Seems like I was going to say something else, but maybe it will come to me. All right, so here's just kind of a little rundown of our course, what we'll be studying. This course has a very clinical bent to it. So what I mean is a lot of you are, you know, future, you know, nurses or healthcare workers. So we've really slanted the course to be thinking most about bacteria and the diseases that they cause. There's a lot more to microbiology. There are lots and lots of good bacteria. There are ones that, you know, break down pollution, oil spills. There are bacteria in our bodies that are necessary for us to be healthy. Try not to forget about them. But we pretty much don't worry about those environmental microbes, the ones in the soil. We just don't really have time to get to those. So again, we're slanted more towards thinking about disease. So when we start out the course here, we're really kind of jumping around a little bit. And the reason that we do that in the book is because we were trying to design the lecture to match up with the lab. And I know that not all of you are in lab. I will only ask you lecture questions. I won't ask you things that, you know, you would have done in lab. Lab definitely helps you understand at least some of these early concepts. Now, because we've had to change lab to once a week, it doesn't match up as nicely. So unfortunately, that's the case. But you know, we're gonna talk probably today yet about culturing microbes a little bit, and next week we'll get into microscopy, classification, really some really fundamental things of microbiology, structure of cells, things that we're going to build on the rest of the semester. The exam dates, I'm usually pretty firm on exam dates. I don't like to change those dates because I know students, you know, schedule doctor's appointments or whatever around them. I may change the amount of content that's included in that exam, but I like to keep those exam days pretty firm. As we get into the second exam material, we start getting into metabolism and bacterial genetics. Sometimes for some students, depending on your background, it's a little bit more abstract, a little bit more difficult. For some students, we'll spend a little time on structure of viruses, of course. And then from then on, we're going to focus for a while on how to kill microorganisms. All right, so what kind of chemicals can we use to kill them? What kind of drugs? How do antibiotics work? And we're going to think about kind of the process of an infection. So lots of terminology, you know, a little epidemiology thrown in there. And then a couple weeks on how we try to kill disease-causing organisms in our body, how our little introduction to how our immune system works. Okay, we have a whole class in immunology that's, I think, offered just in the fall, but this is a little taste of that. And then at the very end of the course, we like to talk about some specific infections. Tuberculosis, anthrax, kind of changes every semester which ones that we do, but I always try to throw in things that are especially applicable at the time. So for a while we talked about Ebola. Of course, we'll talk a little bit about coronavirus. Okay. So that's our semester in a nutshell. Okay, before we jump into lab one, I just wanted to pass on this advertisement. You might have got an email about this through the Class Advising Center. If you are gearing up this winter or spring to take some kind of big test, like you know, dental school exam or MCAT or something, they're offering a once a week online course. I know the Kaplan MCAT course costs what, like $3,000 or something like that. This is like 70. So it might be a good option, you know, if might be something to look into. So I just pass that along. Again, that's the Class Advising Center. Okay, so I try to open up every chapter with kind of a little, okay, this is kind of what we're going to be talking about. They always say public speaking, tell them what you're going to tell them, and then tell them what you told them. So at least try to give you a heads up. Okay, this is what we're going to talk about. So chapter one, a little introduction. What do microbiologists study? A little bit introduction on how to classify microbes. From time to time, we like to look at problems that early microbiologists encountered and how they thought, their thought process, the experiments they did to try to work through those. So fermentation is one of those early things that we'll look at. What causes disease? Of course, we're going to look at that from a historical perspective, but still something that we have to grapple with today. There's emerging diseases all the time. A little bit, some of the important people that helps us understand how to prevent disease. All right, so a little history thrown in here to chapter 1. All right, so let's start out with Antony van Leeuwenhoek. Okay, that's a name that you're going to want to remember. He was a Dutch cloth merchant. Okay, and he is famous for being the one to develop the first microscope. Okay, so the year is not really important there. Just to give you a little perspective, this was early days, mid 1600s, early 1700s. And him being a cloth merchant and developing a microscope, those two things really sound like very different things. But if you think about some of the stereotypes you may have about Dutch people, yeah, they're tall, yeah, they're very clean, maybe they vacuum their house all the time. The other stereotype that you may have is that they're thrifty. We'll say it that way, right? If you go Dutch, then the guy doesn't want to pay for both people. All right, he was in fact very thrifty. He wanted to make sure that if he paid a lot of money for some fine cloth, he wanted to make sure that it was indeed fine cloth. He didn't want to get ripped off. So that motivated him to make these very fine lenses. All right, and he got really, really good at that. So his early microscope here looks nothing like a microscope that we have today. But with his early microscope, he actually had good enough magnification, as good as like the medium and high power lenses on our microscopes in the lab. So he could magnify things like 70 to 200 times, something like that. And it's kind of hard to understand, but this little circle right here, that was the lens. And he would hold that up and look through it. And then there's a little kind of pointy thing here. He would stick whatever he was looking at on that point. So if it was a fly, he would stick that there, hold it up and look through it. Seems kind of uncomfortable, and the lighting might have been a problem. But he looked at all sorts of things, drops of water. And he wrote up what he found and published those papers. And he was a very respected scientist of his time. He was elected to the Royal Society in London there. Okay, so the things that he saw, he described them as wee animalcules. Right? So if we think about the things that he saw and how could we classify those things, those wee animalcules, we could put them into 6 categories. Okay, there's bacteria. Let me get this a little bit bigger. And archaea. Now, we'll talk more about the difference between archaea, those two groups, but those we would call prokaryotes. Okay, no membrane-bound nucleus. He also probably saw some protozoa in water samples. Let's see, some fungi, probably some algae. These are all things that we would consider microscopic. And maybe some other small multicellular, we'll say animals. Okay, so those are the sort of things, obviously, microbiologists look at things that are too small to be studied with the naked eye. Okay, and of course, the other thing that we might include in that group today that microbiologists study, I'll put it in parentheses here because he wouldn't have been able to see viruses because they would have been too small. Okay, we weren't able to visualize viruses till we came out with the electron microscopes in the 1930s. Okay, so we knew their viruses were there, we just couldn't see them. So we advanced our microscopy a little bit. Okay, another contemporary of Leeuwenhoek is Carolus Linnaeus. And Linnaeus was a Swedish botanist And he is famous for coming up with a system to organize all of life. So he's considered the father of taxonomy. Okay, so we're going to talk more about Linnaeus and some of his rules when we get into a little bit more nitty-gritty of taxonomy, but you know, he made this framework so that as we, you know, discover new organisms, there was, you know, somewhere to put them. Rules of naming. He's the one that said we got to have Latin names for everything. Okay, so he helped to kind of organize this life that Leeuwenhoek was describing. All right, so if I give you a few more examples of some of these kinds of life. Prokaryotes. So I said the bacteria and the archaea are prokaryotes. I should add that typically prokaryotes, the cells are going to be smaller than eukaryotes. Okay, and of course this should be reviewed, but of course they're going to lack a membrane-bound nucleus. Of course, they don't have organelles that are membrane-bound, like mitochondria or lysosomes, endoplasmic reticulums. And again, that includes the bacteria and archaea. What's the difference between those? We'll talk about this a little bit more later. But bacteria, they have cell walls where a major constituent of those cell walls, we'll say made of, is peptidoglycan. Okay, and we are going to look more carefully at the structure of peptidoglycan. That's a really important structure because a lot of antibiotics inhibits the formation of peptidoglycan. So cell wall structure is something that we spend a lot of time on. Archaea have very similar cell walls, but their cell walls are never made of peptidoglycan. So I'm just going to abbreviate that and say PG. All right, so archaea may have something called pseudo-peptidoglycan, but it's not the same structure. Okay, only bacteria have peptidoglycan in their cell walls. Okay, we typically think of the archaea as being kind of extreme organisms, like the ones you would find in the hot springs of Yellowstone or polar ice caps or really salty conditions. But there's also archaea that's probably part of our normal microbiome. But we do know that at least so far, we haven't found any archaea that cause disease. So we'll just make a note of that. Do not cause disease. And who knows, maybe someday that will be proved wrong, but haven't found any so far. Okay, so I just put up some examples here. This is an example of an archaea. You can't tell just by looking at it whether it's a bacteria or archaea. This is a kinds of cells that actually produce methane as a byproduct of their metabolism. They're methanogens. Here's a bacteria called Dinococcus. What's special about Dinococcus is it actually thrives in high levels of radioactivity. So I always think it's cool when we find examples of bacteria that really help us. All right, so they help kind of, you know, clean up toxic spills. This picture in the middle here, that's just Staph aureus, Staphylococcus aureus. That's an organism probably a lot of us have on our skin, sometimes pathogenic, an organism that you'll use a lot in lab. All right, so that's a little introduction to our prokaryotes. We spend a lot less time on eukaryotes. There are microscopic eukaryotes. Not as often do they cause disease, so we just don't, we only spend a little time on them. But of course, there's fungi that are microscopic. This one picture right here that's got the kind of golden heads, that's where we get penicillin. So Penicillium chrysogeneum is the name of the mold that actually secretes penicillin. So that's been pretty important in our history. This other picture here is yeast. Okay, Saccharomyces cerevisiae. That's we can thank brewer's yeast for alcoholic beverages. Some example of protozoa. We often describe protozoa by their motility, how they move around. We can describe protozoa as being ubiquitous in nature, meaning they are everywhere, especially bodies of water. Okay, and a few of them are going to be disease causing, but mostly not. All right, so we spend a little time on one protozoa in particular. We don't talk about our algae very much. Of course, they are crucial to the world's food chains. They photosynthesize, right? But they're not they're not ever pathogenic. Maybe the closest example to that would be red tide. This is when we get overgrowth of algae, that the toxins can kill fish and kind of give us respiratory problems. But there's some diatoms there on the right, marine algae or saltwater algae, and a Spirogyra on the left, a freshwater algae. Okay, so those are some microscopic eukaryotes. I'll just mention briefly parasitic worms. This was a picture in your text. They don't even tell you what worm that is. Might be some kind of roundworm. I'm not really sure. We don't really get into any kind of parasites in this class. Of course, those are multicellular pathogens, or, well, usually if it's multicellular, you call it a parasite. If it's a virus or bacteria, you call it a pathogen, if it's disease causing. We do have a parasitology course here at Grand Valley, taught by Dr. Graham. He is a great teacher because he's had a lot of the infections that he teaches about. So take parasitology if you're interested there. And then, of course, the viruses. All right, you may recognize this. This is a picture that I have had on my slide for years and years and years. It's the Sars Coronavirus. Okay, as luck would have it, we're pretty familiar with that now. So viruses can infect animals, right? So we're an animal, okay? So like SARS, viruses can also specifically infect bacteria cells. So we will talk about those and how those reproduce. Some of those look really strange, almost like a lunar lander here. Again, that's a virus that only infects bacteria. So kind of in a nutshell, what's special about viruses? They are acellular. That means they don't have a cellular structure. Okay? Most of the time, we don't even consider viruses as being alive. They're really just a little bit of nucleic acid, and that can be either DNA or RNA, not both. They'll have one or the other with a little bit of a protein shell around them. Sometimes some of them have a membrane around that as well, not all of them. Okay, so they're pretty simple, and we could consider them intracellular parasites. Parasites. That's an E on the end there, parasites. Okay, intracellular, actually I should say obligate intracellular, meaning they have to be inside of a cell in order to reproduce. All right, so we can't put viruses on some bacterial media. They're not going to grow, right? They have to be inside a living cell in order to make more of themselves. And actually, there's some bacteria that have to grow inside of a cell as well. But all viruses, again, they have to be inside a living cell. All right, so that's a snapshot of the kind of organisms we're going to be thinking about this semester. I want to talk a little bit of history here. One name that you're going to hear probably several times throughout the semester is Louis Pasteur. Okay, and some of the things that, or the advances that he made kind of starts around the 1850s. That era of about 1850 to 1910 is often referred to as the golden age of microbiology. So we made lots and lots of advances in microbiology in that time period. And one of the earliest things, Louis Pasteur was a French chemist. And one of the earlier puzzles that he set out to work on was how fermentation takes place. Of course, the wine industry in France was really important. And they were having trouble in the wine industry because they didn't really understand how grape juice got fermented into wine. So when something went wrong, when their wine was spoiling, they didn't know how to fix the problem. So Louis Pasteur came in and he observed that when grape juice ferments, when you look at that fermented product under the microscope, you see both yeast and bacteria. Okay, and really this is at a time where they were really just only beginning to think that maybe these small organisms could be responsible for disease. But he set out and he made a series of hypotheses. Okay, so thinking, at the same time about scientific method here, it's always good to kind of remind ourselves how that's supposed to work. Because a lot of times today, we'll have big companies that don't follow the scientific method. They have some conclusion and they try to get people to, make studies to, prove their conclusion. So we always have to take a look and see, well, who's funding this study? And is that going to bias, what the outcome was? But Pasteur, he had 4 hypotheses that he tried to prove. And of course, you only want to kind of change one variable at a time, a well-designed experiment. His first hypothesis was that, well, fermentation just occurs, like all by itself. Okay, so to test that, he took a flask of grape juice and he sealed it up. He let it sit there for a while. And then he looked at that grape juice, under the microscope, maybe tasted it. Okay, it's still, there's no microbes in there and it's still grape juice. So he could reject hypothesis 1. All right, his next hypothesis was that air ferments the grape juice. And in this case, luckily, he was a skilled glass blower. He made up these special flasks that he called swan neck flasks. And he boiled the grape juice, and then you could see that it would be open to air. But if there was anything in the air, any kind of microbes, those would kind of settle out in the bend of that swan neck. Okay, so he let that sit for a while, and then he found that it was still grape juice, no fermentation, and there was still no microbes in there. So, okay, reject hypothesis 2. It's not the air that ferments the grapes. Okay, is it the bacteria that ferments the grapes? So he sterilized the grape juice by boiling it, and then he added bacteria. He found, after that sat for a while, we got lots more bacteria, but it wasn't really wine. It was a very kind of acidic product. All right, and you're going to find that through lots of experiments if you are in lab, that generally when bacteria ferment a sugar are almost always going to have acidic end products. So we always are testing the pH in lab as evidence that fermentation has taken place. So he got kind of a modified hypothesis there that bacteria do fermentation, but we don't get alcohol from that. Okay, so then he said, well, is it the yeast that are fermenting the grape juice into wine? Okay, and he found that yes, he got lots more yeast when he only added yeast to the wine, and alcohol is produced. Okay, so he was able to accept that. All right, so he came up with this process that he called pasteurization, where he heated up the grape juice enough to kill all the bacteria. They added the yeast back in, and then they would get a reproducible wine product every time without having to worry about whether the bacteria were spoiling it. And of course, we use pasteurization for mostly milk and juices today. I think that they, cut down on bacterial numbers by chemical means today rather than by heating it, because it does alter the flavor of the wine a little bit. Okay, but that was enough to make Pasteur pretty famous. He went on to do a lot of other things. He made a couple of vaccines. He made our first vaccine to anthrax, actually. But one of his other contemporaries that really made some big strides was Robert Koch. And he was a German scientist. Again, he was just a little bit younger than Pasteur, but they were contemporaries. Pasteur was French, Koch was German. Those two sides were at war with each other during the Crimean War during this period. They hated each other. It's kind of sad because they worked on a lot of the same things and they could have collaborated, but that's not the way it worked out. Robert Koch is best known for helping to understand disease etiology. Okay, so that means, that word etiology means causation. Okay, so he helped us understand what causes disease. Okay, and of course, a little bit earlier than this, we didn't really have much idea what caused disease. The prevailing theory was it was bad air. That's where the word malaria comes from. Mal meaning bad, aria, bad air, miasmas. Maybe it was an imbalance of bodily fluids. That's why the strategy was to bloodlet when somebody was ill, put leeches on there. Okay, we now know that that probably just made the patients weaker. Okay, so Robert Koch was very systematic. His experimental design was precise, and so when he came to conclusions, his results were very believable. So he came up with a set of rules to follow called Koch's postulates. Okay, and those postulates, again, were used to help determine what caused a disease. Okay, and he kind of got lucky with the first disease he set out to figure out what was causing. Anthrax was a huge problem, especially in livestock back at that time, and he was the first to really understood that it was a bacterium, Bacillus anthracis, that caused anthrax. And I say he got lucky because it was sort of an obvious one that worked well with his postulates. It's kind of a big rod-shaped organism. It was easy to see under the microscope, and it kind of behaved the way it needed to in order for him to figure it out. If he would have tried to figure out something like smallpox, he wouldn't have gone anywhere because, you know, that's a virus. And anyway, okay, so let's take a look at these postulates. Your textbook doesn't have a figure to go along with this, so I pulled this off the internet. It does talk about Koch's postulates in your textbook, though. So here, 1, 2, 3, 4. These are the postulates. These are the things that Koch said needed to be satisfied in order to prove causation of disease. Okay, so first of all, he said the suspected germ has to be present in every case of the disease. Okay, so in this case, his diseased subjects were mice. Okay, and he sacrificed those animals and maybe smeared out a tissue sample on some media, and then he was able to get each one of those organisms kind of separately, and probably by some trial and error with that, only that organism, he would inject it back into a healthy animal. And if that healthy animal started having the same symptoms as the original disease subjects, and if he could once again isolate that organism from the second diseased animal, then that was enough to prove that it was that organism that caused the disease. Okay, and I should say, he said that germs should generally not be found in healthy subjects. So try as they might, they couldn't isolate that same germ from any organism that was healthy. Okay, so this works well with some infectious disease. Of course, if you've got an infection that only infects humans, if you don't have an animal model, You're not going to be able to satisfy Koch's postulates. If you have a disease that's, say, genetic, obviously that's not going to work. Some infections we know that you can carry when you're healthy. Okay, so that wouldn't really satisfy the postulates either. So there are some reasons that this wouldn't work, but this was a really important first step as people really began to understand, oh, it's those microbes that many times are what makes us sick. Okay, and to go along with Koch's postulates, we had to make advances in culturing organisms as well as staining them. Hans Christian Graham was a worker in Koch's lab, and he developed this really important Graham stain. If you are in lab, you will get to do this numerous times. Hopefully you'll get really good at it. But whenever we're trying to identify an organism, the first thing we always ask is it gram positive or gram negative? Okay, and those are differences in bacteria depending on cell wall structure. So we'll talk much more about that when we get to structure and why some cells stain one color and why some cells stain another color. Okay, in terms of preventing infection and disease, there's several people that we want to remember that really helped with advances in improved hygiene. Okay, that's probably the biggest thing. If you look at drugs to treat infection, vaccines, and hygiene, probably advances in keeping us clean has helped to keep us healthy even more than vaccine and drugs. Ignaz Semmelweis, he was a Hungarian physician, and we're kind of going back a little bit in time here, 1848. Again, this was before they really understood that germs were what often caused disease. Okay, so he advocated hand washing. He recognized in his hospital in Hungary that many women died of infection after they gave birth. They called it childbed fever. And he recognized that physicians that were helping to deliver these babies often came straight from the morgue after doing an autopsy, and they would come up and deliver the babies without even washing their hands. So he thought there might be some particle on dead bodies that was transferred to living bodies and causing this, but he didn't really know what. Nobody believed him that could be helping at all. So he really wasn't accepted very well. He ended up dying in an insane asylum when he was in his 40s. But he must have written up the statistics that he took because Joseph Lister, he was an English physician, and here about 40, 50 years later, he advocated surgeons washing hands in carbolic acid, okay, otherwise known as phenol. Okay, phenol is fairly caustic, fairly toxic to us. So yeah, this did a great job in cutting down infections in the operating room. So surgeons would wash their hands in phenol. They would, wash the patient in phenol before they made the incision. They would spray phenol in the operating room. And this did a great job of cutting down infections. Unfortunately, it had some kind of nasty effects to the people that were around this all the time. So the physicians that bleached their hair white and their fingernails fell off and they had respiratory problems. But again, it was kind of a good step forward. Luckily, we have things that are a little not so harmful to us that will kill microorganisms today. Florence Nightingale, then, about the same time as Semmelweis, maybe a little bit after, she advocated for hygienic conditions in field hospitals. So during the Crimean War, we'll say an advocate of hygiene, okay, clean conditions for soldiers. And what makes really her famous is that she did a great job of, taking data, okay, and she ran the numbers, and she was able to convince people that if we keep our hospitals clean, that we're going to save lives, that there's less chance of infection. All right, and then another branch of microbiology is epidemiology. Okay, we consider John Snow, we call him the father of epidemiology. Of course, the study of how disease spreads. And he is an epidemiologist. Really, it sounds really exciting today, and it probably is, but it also just requires a lot of field work and asking questions and just amassing a large amount of data. Back in 1854, there was a cholera epidemic in London, and they didn't really understand what was causing so many people to have this nasty, deadly diarrhea. So he suspected, though, that it was something in the water. So he went around, he took this map of London, and he went around to every household, and he asked questions. He said, is there anybody sick with cholera? You know, what have you been eating? Where do you get your water? You know, all sorts of questions. And he figured out when he plotted all those red dots on the map there, somebody that had cholera. And then he put in the blue dots are the water pumps. He realized there was a huge cluster of cases around the Broad Street pump. So he took off the handle to the pump so nobody could get water there anymore, and the epidemic subsided. All right, so whenever you have whenever you're looking for a simple answer to a complex problem, you say, where is the handle to the Broad Street pump? Okay, all right. And then kind of lastly, in terms of advances we've made in preventing and treating infection, a couple names to be familiar with there. Edward Jenner, of course, this is going way back, 1796. He is credited with making the first vaccine to smallpox. Okay, and I say credited with, there was probably a lot of other people that sort of contributed to this that don't really get any kudos for it. But he noticed that milkmaids often contracted cowpox. And when they did, they never seemed to get smallpox. So he noticed how these two diseases must be related. Cowpox is a lot more mild than smallpox. Smallpox has about a 30% death rate. And so he took a milkmaid that had cowpox and scraped some of her scabs off and kind of cut a little boy's skin and put some of those scabs from the milkmaid into his skin. He called that vaccination, so the vaca, cow. And, you know, he had this mild case of cowpox. And then he intentionally did the same thing with some smallpox scabs and poked them into the little boy's skin. And luckily, he didn't get sick. Right? It's not quite the way we experiment today, but a lot of good has come of it. Okay, so lots of problems with, you know, infection when you are going to inoculate people that way. Vaccines have thankfully gotten much safer since then. And Paul Ehrlich, he was the first one to really think about how do we treat someone once they have an infection. So, you know, we were making advances slowly in vaccines before that, but what kind of drugs can we use to actually make somebody better? He was famous for searching for what he called the magic bullet. I don't know if I spelled bullet right or not. So a magic bullet would be one that kills your enemy, but not your friend. All right, so he was looking for compounds that when you ingest them, it would kill the bacteria or the virus, but not cause any harm to our bodies. Okay, that's still an enormous problem today. We've got lots of new antibiotics today, but they're all too toxic for us to use. So we need drugs that will target something special about, say, the bacteria and not be harmful to us. All right, and he tested lots and lots of compounds. He was trying to find a compound that would work against syphilis. and he found one that had arsenic in it that was some help against syphilis, but anything with arsenic in it is not really so good for us either. But, you know, yeah, it was the first step, and we've, you know, made progress since then. Okay, so that is chapter 1. All right, whew, I already got 1 chapter down. All right, let's see. Okay. All right, so chapter 6, I kind of split up into 2 parts because I'm taking like just a little piece out of chapter 6 now, again, to try to match up with lab when you're going to be doing your first, starting your first cultures and talking about media in lab next week. And then we'll do the rest of chapter 6 in a couple weeks. So we want to look at now thinking about why do we grow bacteria in the lab? Well, a lot of reasons. One of the reasons, remember, is to try to satisfy Koch's postulates. Okay, so we have to be able to grow them up to kind of prove that that's what's causing an infection. Of course, lots of other things. It helps us identify them, you know, study them. How do we grow bacteria in the lab? Okay, we're going to talk about some medias and things like that. This term, pure culture, that's a really important term, and how do we keep our cultures pure? And then we'll look at some different kinds, some basic kinds of media, and hopefully you understand why that's important that we have lots of different kinds of medias. Okay, so a little terminology first. I usually don't kind of walk through this quite so slowly, but this will be some overlap with lab as well. It's kind of important, so it's okay if you see it a couple different times. But I find that students tend to kind of mix up these words a little bit, so we want to try to use them correctly. That term culture, you can use that as a verb and as a noun. Okay, if we're going to culture something, that means that we're going to grow it up. All right, so we want to culture some E. coli. We want to grow up lots and lots more of it. Okay, if we use culture as a noun, this is where I had my notes written out a little bit better than what probably they're in my mind. The culture is kind of a place where the microbes grow. Okay, so that, for instance, could be the Petri dish and the media, for example. All right, so right here, down here, we have a culture where it looks like bacteria are growing. Okay, it could be a test tube with some liquid media in it. Okay, this term inoculate. If you're going to inoculate a culture, that means that you're going to put microbes in it. Again, I had that written out better in my notes. But you're going to basically start a culture. You're going to inoculate, put some bacteria in it. The inoculum, we use kind of the noun form there. Those are the cells that you're using. Okay, so I'm going to, looks like here in this picture, they're using a loop, which is a wire with a little loop circle at the end of it, to touch some cells on a plate. That's their inoculum. And then they're going to use that to inoculate another culture. Okay. And of course, the medium, that is basically the nutrition for the cells. Okay, so you're the physical, you're growing the cells on the medium. Okay, so in this case, that's the red in that picture there. It looks like blood auger. Okay, plural of that is media. So if you say, oh, there's lots of different medias we could use. Okay. All right, so a little terminology there. Students tend to mix up inoculate and incubate. Of course, if we're incubating some cells, that means we're going to put them at their ideal growth temperature. So for most of the cells that we use in lab, we incubate them at 35 degrees Celsius. Okay, that's really close to body temperature. Body temperature is about 37 degrees Celsius. It seems to be warm enough that most of them grow really quickly. Okay, so we inoculate and then we incubate, wait for them to grow up. So in this picture here on the left, they're using a loop. Probably they dipped that loop in some liquid media with some cells in it, and then spread it over the surface here of this solid media. Now, you can't see any cells there, right? So when you do this next week, it seems like, well, I'm not doing anything. I can't see anything. Have a little faith after you incubate that. If you have, you know, a few, the cells that you put on here that you can't see, they will grow up into colonies. Okay, so they obviously, most microbes were going to reproduce very quickly. Once you get, you know, millions of cells from that one cell, they will be able, you'll be able to see them with your naked eye. Okay, so they have all those nutrients in that medium that they're going to grow really quickly on. Okay, and when we say that there's a colony that's not touching any other colony, we call that an isolated colony. Okay, we're going to talk about why that's important. But it's if you're trying to decide if you have one species of organism there or many, you need to see those colonies off by themselves to kind of compare how they look. All right, so what about the media? Some basics here. So we have both liquid media and we have solid media. Liquid media we usually refer to as broths. Okay, and we have some all-purpose broths that we use in lab. One of them is nutrient broth. How do you make up that liquid media? Well, we buy it in a powdered form like this. You dissolve it in a certain amount of distilled water. You might need like a stir plate, a stirring rod to kind of stir that up and get it dissolved. You heat it up to make sure it's sterile, and then you pour it into whatever container you want. So you pour it into, say, lots of different test tubes. So when you pour that media into the test tube, it's probably going to look pretty clear. Okay, you never know for sure whether it's sterile by looking at it, but if you can see through it, there's probably very few or no cells in it. The cloudier that media gets, the more turbid it gets, then the more cells that you have growing in there. And sometimes if it sits for a while, you're going to have those cells settle out to the bottom, and you might need to kind of stir that tube up to be able to see it again. Okay, so the cloudier, the more cells are growing there. But we tend to use liquid medias. They are good for growing lots of cells quickly. Okay, so they've got the cells have lots of room to move around. You put them in a nice warm place, lots of nutrients. Generally, they're going to grow really fast. The drawback here is you can't look at that tube and understand, well, is there one species there or are there 20 different species? You have no idea because they all look the same. Okay, so that's the nice thing about solid media. Different cells will take on a little bit different appearance. Okay, again, so solid media, we'll say good for getting pure cultures. I haven't defined that yet. A pure culture is when there's only one species growing. Okay, so generally in lab, we like to keep our cultures pure. We like to be working with one organism at a time. Okay, if we work with more than one at a time, then we can get goofed up on what's doing what. Okay, so we generally like to keep our cultures pure. How does media get solid? Well, it has Auger in it. Okay, Auger is a solidifying agent. So basically, we can take some nutrient broth, add some Auger in a powdered form, heat it up, dissolve the agar, and then when it cools down, we can pour it, well, we pour it into, like, say, a Petri dish here or a test tube. The test tube, we typically let them cool on an angle, so we get a slant here. So our solid media, we have slants and plates. We can pour that, again, media with agar into it, and when it cools down enough, it will stay solid. What is agar? It's actually a polysaccharide, which is derived from the cell walls of algae. We'll say seaweed. It's actually a red algae. Okay, so what's really, really important about agar to understand is that the bacteria are not eating the agar. Okay, it is inert. It is to say that another way, it is not a nutritional source. Not nutrition source. Sorry, that's really kind of sloppy written up there. So if the bacteria ate the agar when you were incubating it, your plate would all turn to mush. That's not what we want. We want it to stay solid. So the nice thing about agar is it's this polysaccharide that the vast majority of bacteria cannot metabolize. Okay, think about another polysaccharide, cellulose. That's in green plants. That's why we can't eat grass and get nutrition out of it. We don't have enzymes to break down cellulose. Okay, so again, most bacteria are not able to break down agar, and that's what we want. Where does agar come from? I just included this here. Oh, there it comes. We actually have to collect it. So after storms in places like Morocco or Spain or Korea, people go out and collect the seaweed that washes up on shore, and then I make that into a powdered form of agar. And they first started using agar. Robert Koch first started using it in his labs back in the 1880s. We haven't found anything better since then. So we're still using the same thing. We haven't been able to cultivate the seaweed. So a few years ago, I got this from an article in Nature. They were worried that we were over farming it and there was going to be a shortage and the price of agar was going to go way up, and that never really seemed to pan out. But okay, so how do we get pure cultures in lab? Remember, so a pure culture, one species at a time. Usually the method that we use, we streak our bacteria onto some kind of agar media. Oftentimes what we'll do is called a streak plate. You're going to do lots of these in lab, but it's a special technique that we use to try to spread the cells out, dilute them out on the plate until we get isolated colonies. Okay, so you start out, you streak kind of in one quadrant, you flame your loop, you clean off your loop, okay, and then you spread it out a little more, you clean off your loop till eventually, hopefully, you get lots of isolated colonies. And if all those isolated colonies look the same, you can be reasonably sure that you only have one species on there. Okay, another method that we're going to use in lab is called a spread plate. It's not quite as foolproof for getting isolated colonies, but you would put maybe a drop of liquid media with cells on it and spread that around with a glass rod. You'll have a chance to do that if you're in lab. Okay, and then we're going to try to keep our cultures pure by something that we call aseptic technique. And your lab professor, again, if you're in lab, we'll be harping on you about this. Lots of things we do to try to keep our cultures pure, because it's very easy for something to float in here and contaminate the plate, something that we don't want growing, because there's all sorts of microbes floating around in the air. So we're going to, for example, keep our lids on as long as possible instead of leaving your plate open to the air. You're going to disinfect your bench top just to kind of cut down on the chances that something growing on your bench would get into your culture. You're going to wear a lab coat. You're going to flame your loop. Like, there's all these things that, again, I'll leave that to your lab professor to explain that a little bit better. But we almost always want to try to keep our cultures pure. Okay, so here I'll leave off with this. This is what a mixed culture looks like. Okay, you can tell that there are many different organisms growing here. They all have a little different appearance, a different edge, a different color, a different size to the colony. So sometimes it's really easy to tell if you've got lots of different things growing. Sometimes it's a lot harder than that. Okay, so we will pick up there next time. If you have any questions, come on up. Thanks for coming.